Hey class, hope you guys are doing good. All right, let's do our Unit 3 test review. So these are the questions that you guys sent in. Thank you guys for sending them. Hopefully this is helpful. All right, so let's go through Chapter 19, the immune system. How does a bacteria affect your body? So this is a slide where the answer is, and the part I want to highlight is in bold. So what bacteria do is they affect the body by carrying or releasing toxins which can disrupt normal cell function or straight up kill cells. Um, so just the cell wall of bacteria has toxins that are harmful for the human body. So just them, if you get enough number of bacteria in your system, that can cause toxicity, and it can potentially kill you, um, or it can cause irritate cells in your digestive system and cause you to have diarrhea or things like that. But those toxins damage cells in your body. How do RNA viruses infect cells? So here, the RNA viruses part, I just say RNA viruses because that's what's in the figure of your textbook. So this is the example they give of how a virus attacks. And I just want you guys to know, to recognize this figure, and to know the general flow here. But what's happening in the first step is the virus is binding to receptors on the surface of the cell, which are like locks, and the virus has the keys. There's a video on the next slide that I'll show you that talks about this. But anyway, the virus can get access into the cell, and it releases its genetic information into the cell. And so the second step, in this case, the RNA has to be reverse transcribed into DNA. So this happens with an enzyme that does the opposite thing as RNA polymerase. So you know that transcription is when you make a copy of DNA as RNA. This is the opposite. RNA is reverse transcribed into DNA. Then the third step, that DNA, now the cell just thinks it's its own DNA, but it really contains the genetic information for the virus, and it transcribes and translates this and makes viral protein. And these proteins can assemble together. So the fourth step, these proteins and the RNA assemble together to make new virus, which are released through the membrane and can infect other cells. And so this is a video that I showed in class, and I recommend you guys watch that again. It animates that whole process for you guys. Okay, what kind of cells characterize innate immunity, and what type of cell bridges the innate immune system with the adaptive immune system? So this is the summary slide of innate immunity and adaptive immunity. And you can see the first question, what kind of cell characterizes the innate immune system? Under that innate immunity bracket, there's two kinds of cells here, macrophages and dendritic cells. There's also other cells that are in this innate immune system, neutrophils. Um, really, those are the only three worth mentioning, macrophages, dendritic cells, and neutrophils. So what these cells do is they eat. They do phagocytosis. So they will eat bacteria or viruses or whatever it is that's infecting the cell, that the cell doesn't recognize as part of the body. And so the macrophages and dendritic cells will eat the pathogens. Then what type of cell bridges the innate immune system with the adaptive immune system? That would be the dendritic cell in blue there. So the dendritic cell, what it does is it eats the virus or the bacteria, chews it up, and then presents antigen of that pathogen on the surface of the cell. And it presents it to cells in the adaptive immune system, to B cells and T cells. So basically what it's doing is it's saying like, look, this is what the virus or the bacteria looks like, and then the B cell can create antibodies that will recognize the pathogen and help fight the infection. Or the T cells will create receptors for the antigen so that they can recognize infected cells. Okay, and then how do vaccines protect the body from viral infection? So here's the slide. The bolded part really that I want you to know is that the T cells and B cells that have been exposed to the vaccine, which really is just the chewed up uh, virus or bacteria. So it has viral or bacterial antigens. So the T cells and B cells that recognize these antigens have long term memory that can recognize the antigen decades later. So if the virus infects you again, the T cells and B cells are like, hey, we know this is a virus because of the vaccine, so let's fight this infection. So basically, your adaptive immune system is primed to attack. It's like on an on state to attack the virus when it infects. Okay, the nervous system. What is the myelin sheath and why is it important? So here in this slide, um, we're talking about speeding up the action potential, and you can do that by the axons or neurons do that by the myelin sheath. So myelin sheath are these marshmallow-looking things that are really Schwann cells that are wrapping around the axon, and so they form these little marshmallows. And so what they do is they allow action potentials to jump from node to node. So the node are the unmyelinated patches, the parts of the axon that don't have these myelin sheaths. And so the action potential jumps from node to node. And so really these myelin sheaths allow the action potential to take shortcuts, right? To jump. So if you take away these Schwann cells, these myelin sheaths, then you would slow down the action potential, right? Because now the action potential can't jump from node to node. It has to go throughout the whole axon. And what is the propagation of an action? Oh, sorry. They should say, why is the propagation of an action potential compared to a domino effect? So in this slide, we talked about depolarization, but here I just want you to focus on the figure. So basically what's happened here is that an action potential has started and it's traveling down the axon. So the sodium channels are going to sense the action potential and they're going to open up. And as they open up, they let sodium ions in, those Na plus in green. So they let those sodium ions into the axon, and that's going to depolarize it, change the polarity of that axon. And so the next set of sodium channels are going to sense that depolarization, and they're going to open up and let a lot of sodium in. And that's going to depolarize a membrane. Basically, when I say depolarize, what that means is that the inside becomes more positive than the outside because you're letting in all these positive ions. And then the next sodium channel is going to sense it, open up, and let more sodium in. So it's like a domino effect. And I suggest you guys look at animations of this. So even if you just go on Google and type in animated action potential, you'll find some cool videos on that. There's a helpful animated image on Wikipedia. So if you just label action potential animated GIF, that's a word, you'll pull it up and it just shows you how the action potential moves down the axon like a domino effect. All right, last questions. What is a process called by which neurons communicate using neurotransmitters? And what kind of proteins do neurotransmitters bind to in the dendrite of the receiving or postsynaptic neuron? All right, first question, what is the process called? That's actually bolded on the last paragraph there. The process of relaying information from one neuron to another is called synaptic transmission. And that's what's blown up there in the figure. So in the figure, it's showing you that what happens in synaptic transmission is that vesicles that contain neurotransmitters are released once the action potential gets to the end of the neuron. The end there is called the terminal bouton, and so those vesicles move down to the edge of the terminal bouton and release their contents into the synapse. And then those neurotransmitters are going to bind to receptors, and the impulse begins in the other neuron, the postsynaptic neuron. So there you can see that um, the the synapse is a space between the terminal buton and the dendrite of the postsynaptic neuron. So the other question was what proteins do the neurotransmitters bind to in the postsynaptic neuron? And that those would be receptors, neurotransmitter receptors. So what that's going to do is once the neurotransmitters bind to these receptors actually are going to open up and let sodium ions in, just like the receptors on the axon. And so that's how the action potential is started, is right here. Okay, all right, well, that's the end of the review. Hope this is helpful, and study hard. Hope you have been studying hard. And I apologize that I'm sending this in so late, but I got the questions in a little late and had a pretty busy weekend. But hopefully you guys still get to enjoy this and find this useful. All right, see you guys on Tuesday morning at 8 for our exam. Take care. Bye.